Okay, everyone, so this is the first PowerPoint, or half of the first PowerPoint in Unit 4. We are going to be starting drug therapy, drug resistance, things of that nature, and this first particular part of the PowerPoint is absolutely not difficult, so enjoy it. When you see the word chemotherapy, right away everybody thinks of cancer, but in fact, chemotherapy is treating people with chemical agents, which are medications, which is why a chemotherapeutic agent, by definition, is drugs used to treat diseases. There are two scientists that you need to be familiar with. One of them is Ehrlich. He was a German scientist that was working with treating syphilis, yet he was using an arsenic compound, which unfortunately caused him to kill off a whole lot of his patients along with the syphilis. The second scientist that we're going to talk about is Fleming. So Fleming is the founder, essentially, of penicillin. We talked a little bit about penicillin in, I believe, Unit 1, and penicillin is, in fact, a fungus. So he had discovered one of the most popular, widely used to treat bacterial infections, which is the antibiotic penicillin. So chemotherapeutic agents, again, please don't think of chemotherapy as just related to cancer, fall into different categories. When you talk about antimicrobials, this is another way of saying antibacterial, which also is an antibiotic. As you know, antibiotics do not work for things like viral or helminthic infections, and therefore, on the other side of this little flow chart here, we've got other types of medications. So whether it's someone taking Lipitor for cholesterol, insulin for diabetes, or antiviral or anti-cancer medications, that is just one other type or category for chemotherapeutic agents. We have to look at the spectra of drugs because it's very important that people understand the difference between a narrow spectrum drug versus a broad spectrum drug. If a patient shows up in the emergency room and they have relatively general symptoms, then often a physician will treat that patient for whatever ails them if they're able to visually determine what's going on. But more times than not, tests have to be run, and then those test results have to come back in order for the doctor to narrow down and know exactly how to treat them. A broad spectrum drug is a type of drug that will cover various different types of infections based on the basic symptoms. So in a situation of an emergency, more than likely that's going to be administered first. Once the test results come back and the physician is clear as to what illness the person has, then they would go for a narrow spectrum drug, which is specific to actually treat a certain illness that has been diagnosed. If you take a look at this chart, this shows the spectrum of different antibiotics. And if you take a look right here at erythromycin and tetracycline, both of them cover gram-negative bacteria, gram-positive bacteria, and chlamydia and rickettsia. Now, at the same time, that would be a broad-spectrum drug that the physician would give to the patients. Once it comes back, if it's known that that person has a gram-positive bacterial infection, then these two wouldn't necessarily be logical. It might be smarter to stick to something like penicillin, because if you look at the chart, the majority of this falls under gram-positive bacteria. Now, let's say a patient is allergic to it, then of course you would go to an alternate medication, but narrow is after drug results for the patient's illness have come back. So that's broad in spectrum. And then you also have to consider the effects of the drug on the person's body. So if something is a bacteriostatic drug, it will just lower the levels and keep them at a flat line if you were looking at it on a chart. Whereas bactericidal, think of suicidal, that will actually kill off the drug. A good example here to use is our gut and our GI tract. We have E. coli as normal flora. So if someone has a foodborne E. coli infection, you don't necessarily want to treat them with a drug such as a bactericidal that will kill off all of the E. coli, because some of that is healthy normal flora. You would use something that is static so that the drug maintains the regular normal flora levels and then kills off the E. coli that is actually pathogenic from the bad food that was ingested. If we were in class right now, I would ask you, if a person had a gram-negative bacteria, would you prescribe them with penicillin? Because penicillin does fall under gram-negative, and hopefully you would all answer no and say that polymyxin would be a better suggestion because that fully falls under gram-negative bacteria, making it a better drug to treat the infection. Couple of comments about what antibiotics need to be and what they need to do. First and foremost, it's incredibly important that the antibiotic be toxic to the pathogen to kill it off, but non-toxic to the host. Last thing you want to do is kill the host along with the pathogen or bacterial infection you're trying to treat. It has to be able to reach the site of infection. So if you have athlete's foot, more than likely you're not going to put a lotion on your nose or on your arm. It will go directly onto your foot. A lot of drugs are administered intravenously or intramuscular. If that's the case, it's usually a little bit stronger. Topical is not always the way to go, but you wouldn't give topical for something that has to be ingested in the form of a pill. It has to be hypoallergenic so that the host doesn't react negatively and have a hypersensitivity or allergic reaction to the antibiotic. So the next one is pretty similar, effective against the host, but no resistance. So we're going to cross this guy out and say effective against the bacteria, but that it not be resistant in general. And finally, not readily metabolized by the host. What does that one mean? One of the most common calls to pediatricians and emergency teledoc lines for people that have babies and young children is, my child is ill, I gave them the medication, and within 20 minutes they vomited. So now, do I redose my child or not? And the answer is traditionally no, because some has already been broken down. Many things that we ingest in the form of pills are not readily metabolized. For example, if you take multivitamins from, say, Costco, they have a deep yellowish orange color, and then when you urinate, you notice in the urine it picks up the color of that pill because instead of utilizing it in the body, a lot of it was excreted during the process of urination. So I had mentioned this when we talked about the outline review of outline for Unit 4. There are 5 different modes of action of antibacterial agents. What that means is there's 5 different ways that drugs tend to work to kill off an infection. And again, think back to the previous slide. You want it to be non-toxic to the host, but at the same time toxic to the pathogen that's causing the infection. We're going to go through these over the next 4 or 5 slides individually, but essentially those 5 are one that inhibits cell wall synthesis, another one that inhibits protein synthesis, and again, this is in the bacteria causing the infection. Something that disrupts the cytoplasmic membrane, that would be like a MAC attack. Something that inhibits a metabolic pathway. I love talking about this one. These are sulfa drugs. And the last one is something that inhibits DNA and RNA synthesis. So it basically stops the bacteria from making genetic material in order for it to be more effective in dying off. If you take a look under each one, there are examples. On your Unit 4 exam, you need to know all 4 of these in a simple sentence of what it actually means, and one example for each of the 5. I've written many examples, so if you take a look under inhibition of cell wall synthesis, not many of you have heard of possibly cephalosporins, but most people have heard of penicillin. So try to remember one that you're already familiar with to make it a little bit simpler for you. So again, we're going to break down the 5 on your unit outline. There's an area on the first page and the top of the second page where you can take little notes for yourself. On each of these, you'll notice that I'm actually giving you examples. So for this one, this is inhibition of protein synthesis, and the 2 examples on here are, one is tetracycline, and the other is streptomycin. You need to know one for each. So basically what happens here is you have protein synthesis occurring. This is to make polypeptide chains. This is essentially the steps when we're talking about gene expression. And what happens if you have your tRNA coming in, AUG, if you remember from Unit 2, is your start codon. And then this guy would transfer on to the next one and then read CGC. And up here would be GCG. But instead, the drug tetracycline, which is this yellow pill-looking thing right here is blocking it. So now the tRNA can't go further and it inhibits the rest of this protein from being synthesized. So again, inhibition of protein synthesis. The drug, in this case, could be tetracycline or streptomycin, acts like a blocker, sort of like the repressor when we talked about the lacoperon, which prohibits the tRNA from allowing it to do protein synthesis. An example of another one is inhibition of nucleic acid synthesis. The best drug that I think is simple to remember is quinolones. If you Google it and look that up, more of you than you realize are in fact familiar or have heard of this drug. What this guy does is it looks, NT, right here, let me just write this out for you, are nucleotides. So this group of antibiotics, the way they work is they are structurally similar to a normal nucleotide. In other words, we all know that A pairs with T and C pairs with G. So if we were to go further, A to T, C to G, and if quinolone would come in, it would almost take the place of what would have been the A or the adenine, and therefore it stops the rest of nucleic acid synthesis. So it quits and prevents all of the processes of replication, which then stops all of gene expression from continuing on. Next, we have inhibition of cell wall synthesis. Examples for you are penicillin or cephalosporins. Most of you, I assume, are comfortable with penicillin, so definitely remember that one. But what happens here is that this particular antibiotic stops the cell walls in the microbes. So it kills off peptidoglycan synthesis. This is very effective because eukaryotic cells and our cells in the body that essentially are hosting this bacteria causing the infection, we don't have peptidoglycan in our cells. So these group of drugs are effective in killing off the bacteria and rarely then harming the host, because what it's targeting is, again, this peptidoglycan that we don't have in our own cells. Next one, and as I mentioned, this is absolutely one of my favorites. It's interference with metabolic reactions. So this guy works under the realm of competitive inhibition, just like when we talked about enzymes and feedback inhibition. So folic acid is something that most people are familiar with, especially pregnant women, because they are in fact told to take folic acid when they are pregnant, and they take usually 750 to 1,000 milligram or mcg pill. In order for us to consume that much folic acid, we would have to eat pretty much a 5-gallon bucket from Home Depot full of broccoli every day. So as we talked about metabolically losing some of these pills through our urine and when we excrete urine, in this case, of that 1,000, the whole thing doesn't get absorbed by the body, but we do ingest the pill during pregnancy anyway to ensure that those levels are there. Humans don't need it and don't need it to survive, but it is an important component to intake during pregnancy. So the difference is that for humans, it's not mandatory, but what's unique and what you need to understand to make this make sense is bacteria actually require folic acid and die without it. So folic acid. And the drug that we're going to talk about are known as sulfonamides. The easiest way to remember it is its nickname, which are sulfa drugs. Anyone work in the dental field, you'll see a lot of people are allergic to this particular drug, and which is unfortunate because this is a wonderful one where it's almost guaranteed to not impact the human, but in fact just affect the infection. So in a normal situation, this is normal folic acid production in a bacteria. There's an enzyme. All the substrates are binding into the active site, and then synthesis occurs, and the bacteria makes folic acid. This is occurring in an ongoing fashion because, again, bacteria require this to essentially survive. So then when someone has an infection and takes a sulfa drug, this right here, this little rounded corner is the sulfa drug. And notice how up top here, everything nicely fit into the active site. But notice what happens. In comes the G, in comes the P, or it tries to, but this sulfa drug, because of this angular shape, is acting as a competitive inhibitor, and therefore there is no synthesis, and folic acid is not made, thereby killing off the bacteria, because again, they require folic acid to be made for them to successfully replicate. Then one of the last one is inhibiting cell membrane function. So this is another group of drugs that wonderfully are not toxic to us, whereas they're toxic to the organism that is causing the infection. The example I'm going to have you use is polymyxin. This is found in things like triple antibiotic creams, Neosporin, bacitracin, any of those antibiotic creams. When someone skins their knee, and right away you put that on there. So the reason this works is because within, look right down here on the bottom right, this is a cell membrane. When we talked about the components of our cells, we always talked about a cell membrane, and I had mentioned how there is cholesterol embedded into the membrane. And the cholesterol, when the membranes push together, they make them almost flexible and bouncy, because if they didn't, every time we poked at our arm, it would be a permanent indentation. So in this case, these particular group of drugs actually inhibit something, look on the bottom left, known as ergosterol. So in eukaryotic cells, which is us, and again, we're taking this drug or using this ointment to combat an infection, our cells contain cholesterol. But bacteria contain something called ergosterol. You should notice there's a sterol at the end of this one and a sterol at the end of this one. So they are, in fact, in the same group, but at the same time, they are not the same. So you take this particular medication, and it causes holes in the cell membrane by looking for this ergosterol. So it causes a MAC attack based on this sterol that is only found in bacteria, thereby killing them off. Tuberculosis is traditionally the illness that is used to talk about drug resistance. And why? Because if you look on this picture alone, and this is not something you need to memorize, this is just to show you that oftentimes illnesses need combination, or what we refer to as multi-drug treatment. TXT is short for treatment. So here, there's a drug that prohibits cell wall synthesis, or disrupts metabolism, or disrupts RNA synthesis. So the list goes on and on. Many times you cannot treat a patient with just one drug, but in fact you need to give them a multitude of many different ones. This picture covers the five modes of action of antibacterial agents. This picture also appears separately on your outline. So what I'm going to ask you to do is look back at the last 5, 6, 7 slides, write a little bit of information, write an example of a particular drug in that group that falls under that type of mode of action, and that will ensure you that you have in fact understood it and studied it well. So here we have injury to the plasma membrane. This on the earlier slides is written as a cell membrane. Polymyxin is the example, and again, what this does is it combats ergosterol. So it is wonderful against that particular sterol in the membrane, because eukaryotic cells such as ours have cholesterol, and this doesn't combat that. Then there is inhibition of synthesis for metabolites. This was your sulfa drug. Yet again, you have a drug that is combating sulfonamide, which is mandatorily needed for the survivor of the bacteria for their normal metabolism, but not so much for humans. And then tetracycline and streptomycin are your examples for inhibiting protein synthesis. This is the one where it blocks the tRNA. It almost looks like a tic-tac laying sideways on that mRNA strand, and by pathogenesis, it prevents protein synthesis in the organism causing the infection. Then we have inhibition of cell wall, which are things like penicillin and cephalosporin. Some of you might have heard of vancomycin. So in this case, again, it's going for things in the cell wall like peptidoglycan, which is wonderful for eukaryotic cells because we don't have something like peptidoglycan. And the last one is inhibiting nucleic acid replication, which then in turn takes gene expression, because again, if you don't have DNA replication, you can't have any other processes, and it completely shuts that off as well. Quinolones is the example you had for that. So please take a little bit of time to look over this slide and label it, as well as the writing on the beginning of your outline, and then you will have this stuff pretty well understood.